So our next speaker is Connor O'Hara Baker from the Elkhorn Slough Foundation on the terrestrial side, now looking at plants.
Thanks, Kirsty. Good afternoon, everyone. My name is Connor O'Hara Baker. I'm a conservation projects manager with Elkhorn Slough Foundation. And today I'll be presenting about ESF's efforts to quantify these battery cathode metals in upland plants around the Elkhorn Slough region, so above the tide line. So the question we set out to answer is what concentrations of these metals, specifically nickel, cobalt, and manganese, can we detect in plants in the Elkhorn Slough region following the Vistra fire? And are there any particular locations or vegetation types that are associated with higher levels of metals? So in March 2025, 2 months after the fire, ESF staff went and collected 77 plant samples from 15 of our properties around the slough, as well as one control site in Santa Cruz County. A sample consisted of 50 grams of material, and these were bulk samples, so combined shoot tissues, so stem and leaf combined, and 13 samples also included root tissues. Down here on the left, you can see the 5 different veg types that we collected, as well as the number of samples per veg type. Ruderal here refers to subwoody plants like mustard or radish, and then wet refers to wetland perennials like rushes and sedges. We then sent the samples off to an accredited lab to perform ICP-MS analysis, which you've heard about already. They performed pretty standard sample prep procedures, and they quantified nickel, cobalt, and manganese, running duplicates of all the samples and reporting both of the test values as well as the relative percent difference for each sample. And they used 8 processing blanks to sort of deal with potential background contamination. So we saw quite a high degree of relative percent difference between values in samples, and we suspect this had to do with the combining of tissue types in the samples, because we were trying to mix stems and leaves and then even roots in some cases. Even blending them all together, they just couldn't completely homogenize, which leads to, the sample having higher or lower pockets of metals. Down on the right there, you can see kind of the distribution of values for each of the 3 metals, and each has kind of a pretty distinct range. So these are all of the values separated out by metal and vegetation type. And you can see that the majority of the samples have cluster into pretty low values. And then there are around 4 to 6 highly elevated values per metal also. And these tended to be in herb samples, ruderals, and tree samples. And there is one shrub, I think, in there as well. That was an outlier. So like other researchers have already presented, we found strong evidence in this 2-to-1 mass ratio between nickel and cobalt, indicating a common source. This matches surface enrichment levels that Ivano found in the slough. And I just also want to point out how much higher the Elkhorn samples how much higher some of those levels are than the control samples. There were only 7 control samples from one property, so the trend itself, the correlation between the metals is harder to make conclusions about, but none of those values in the control samples were anywhere near what we saw in the Elkhorn samples. Another interesting observation was that roots significantly affected the levels of nickel and cobalt concentrations. So root samples that included roots tended to have much higher values of these metals. And it's hard to say exactly why. This might have been because of some uptake or adhesion mechanism below ground, but it also very well could have been caused by soil particles clinging to the roots because our samples were not washed. And I'll talk a little bit more about that later. Another thing we wanted to find out was whether there were any hotspots in the area. So one simple way we thought to do this was to create a simple index that would combine the 3 metals and identify any points that had a high combined signal. And we did identify a few high combined signals, namely some grass in a cow pasture, some telegraph weed at Sandhill Farm, which was kind of in direct northeast line of the plume, and then an oak leaf and stem down kind of directly east from the fire location. Looking directly at nickel, there were elevated levels in a few of the samples that exceeded plant toxicity threshold of 10 parts per million. And that's for sensitive plants. Tolerance ranges vary quite widely. But another interesting thing we found was that vegetation type did significantly explained a significant amount of the variation in nickel concentrations. Turning to cobalt, it's a similar story. There were elevated levels in some of the samples that go above the normal healthy range of most plants. And veg type also significantly affected cobalt concentrations. Manganese was a slightly different story. So although there were some elevated levels of manganese is an essential plant nutrient, and there's and possibly even a wider tolerance range for manganese among different species of plants. So normal is sort of hard to define, but I found this range cited 50 to 800 parts per million, and all of our samples fell within that range. But again, vegetation type did significantly have an effect on manganese concentrations. So moving on to interpretation of this analysis, some caveats are important to highlight. We like others have stated, baseline data would have been really valuable. We didn't have baseline data on metal concentrations of plants in the watershed. We lack an understanding of the pathways of potential harm to humans and wildlife from contaminated plants specifically. We had honestly too few control samples and uneven veg group sizes, which hindered our statistical power. And that was a practical consideration of, like others have said, ICP-MS is very expensive. We bulked plant material again for cost reasons. And this led to the poor homogenization and kind of unstable numbers. And lastly, we didn't rinse our samples. Unfortunately, I knew hardly anything about this whole process before doing this project. And the lab did not rinse samples as part of their standard procedure. So as a result of that, we can't tell whether metals were inside the plants or clinging to the outside. If I could do it again, I would probably do half and half to compare those. But we did learn some valuable lessons from this effort. Elkhorn vegetation did have elevated nickel and cobalt levels relative to the SoCal control site. Highest manganese values were found in tree samples. We found that very tight 2-to-1 correlation of nickel and cobalt. Samples with roots tended to have higher concentrations overall. We highlighted a few hotspots in the Elkhorn Slough region, and vegetation type had a significant effect across all 3 elements. And with that, thanks for listening. Thank you, Kirstine, for your help with the analysis, and I can try to answer any questions.
Well, Connor was a bit hesitant to speak because it is sort of a preliminary study, but to my mind, all these different pieces put together paint a really thorough picture of what happened. So, it's by combining everything we know. And even if you don't know if those metals were on the outside of the plant or in the inside, it's part of the story of those metals being in watershed. So I appreciate you being willing to speak about it. Questions?
Yeah. Thanks. I think lessons learned is super important too. So I really appreciate you sharing, if I could do this again, that's incredibly helpful. Did you anecdotally notice any decrease in vigor of plants, discoloration, anything while you've been out and about since, or?
No,
Okay.
no, honestly, I was keeping a lookout more last year after this and during sample collection. Didn't really notice any reduction in vigor or anything that looked like, I'm not an expert, but I didn't notice anything like that.
Yeah.
I was curious, of the samples that you included roots for, and you expressed concerns about soil adhering to the roots, could you give a picture of how big is the zone? Is it a taproot situation? Is it filamentous roots? Is it a lot of soil? Were you shaking it off? Because that could still be the plant retaining sediment.
Yeah, totally. So I chose to do that comparison with herbs just because that was the veg category that I had the most samples in. And so herbs tended to be filamentous and not taprooted for the most part, sort of finer roots. And I think I did shake them or brush them off before bagging them. Does that answer?
Yeah, it's just because that would make me think that the stuff that didn't just immediately fall off is at very least something the roots are holding on to. So I still think that's meaningful.
Totally. Yeah.
We're in the midst of a campaign of collecting pickleweed from all around the estuary because we've learned it's a known bioaccumulator of metals. And Emma is the one who's organizing washing the roots. And
Everyone's got tips.
She needs all the help she can get if you want to come wash pickleweed roots. And it's
It's like a hair salon for pickleweed.
Yeah, Connor, I think, when trying to remember when we started this research that you did, one of the things that concerned us was we were planning to potentially do with the prescribed fire burn in this area. And one of our questions was well, is there going to be a bunch of these battery materials in the plants? And then we light them on fire again. And do we re-volatilize these constituent metals, or what's going to happen? Have we gotten any closer to answering that question, do you think?
Through this?
Just in your research in general, right? Been looking into this for a while, yeah.
Yeah, well, I would love to do this again if we had the resources to do it. And particularly focusing on the area that we're hoping to burn. Because this was last March and a lot of these, I mean, the annuals, the rudderals, a lot of these species, I suspect would either not be, or the individual samples would have turned over, and I think it would look different. So yeah, I don't have any like time series to say where, if there's a trend, but it would be great to do it again.
Any questions on Zoom?
Yes, I have a question then, and this is because we were doing a deep dive into how we would wash our plant samples.
Mhm.
And one of the papers we found talked about the hairiness of the leaves. Do
Yeah.
you know enough about the plants that you collected, like, to think whether the hairiness mattered and how much got retained in the samples you got? Like, could that be a factor?
Well, I remember seeing a how do you say that word? Microscopy image that from Ivano, I think, that showed particulates adhering to the outside of, I think it was a bristly ox tongue plant that's really hairy and warty.
Speaking, and I was washing it and go away.
And it wasn't going away.
Experiments here, washing, measuring, washing, I think it's, yes.
Yeah, that's interesting. I mean, out of the species we collected, there was a wide range. There were some that were like waxy cuticle. There were some that were pretty hairy. That would have been an interesting thing to track as a factor. But
Well, one of those plant species you mentioned that had high enough was telegraph, right?
yes,
This is quite hairy.
yeah, it is. Yeah, so that's potentially a reason why it captured more of that. Because also, is something interesting is that I think between the fire and when we collected in March, we did get at least 5 inches of rain in the area. So that, you know, not saying that the rain would have washed everything away that was able to be washed away, but
I
yeah,
mean, that might be the pathway by which your roots were enriched, right? That your plants with
Yeah.
roots have more got into the root zone.
oh, yeah.
All right, then I think we'll swap out the presentation. Thank you.